Hello, students. Let us start with a very interesting topic proteins from chapter 6 biomolecules. What are proteins? The term protein for the first time was derived by a Greek word from a Greek word proteus. The meaning of this word was primary importance or of first importance. This term was derived by Jacob Berzelius in 1830. Later, Gerardus Mulder, he referred these proteins as complex organic nitrogenous substances. He gave the chemical composition of protein that they contain nitrogen and they are present in both plants and animal cell. They form about 10 to 15% of the total cellular pool. So 10 to 15% components of a cell are nothing but protein. So Berzelius derived the term protein and Mulder was the person who told that proteins are made up of nitrogenous compounds, nitrogenous substances. Let us today see the characteristic features of proteins. These are large molecules, macromolecules containing about 100 to 3000 polymers, that is chains of amino acids. The smallest one known is ACTH, adenocorticotrophic hormone. This is a hormone which is released by pituitary gland and it is made up of, like, the molecular weight is 4500 Dalton. Whereas the tobacco mosaic virus, I hope you remember the structure of a tobacco mosaic virus, the tadpole shaped virus with hexagon. It has a protein coat, and this protein capsid, the protein coat of this tobacco mosaic virus, it is about 40 million Dalton. So this protein coat, it is made up of 40 million Dalton. Let us see how a peptide bond is formed. Now students, listen carefully. This is the first amino acid. Let me use some other color to explain you. This is the first amino acid. This is the second amino acid. Now any amino acid, basically it consists of a carbon compound in the center. Carbon has valency 4. At first bond, NH2 group is present, which is the amino group. At one end it may have COH group, that is acetyl group. It has hydrogen at one end, and at other end it may have any R group. It can be methyl, ethyl, or any organic compound. Any R group is present. Similarly, this is second amino acid. You can see again here it is NH2 group. Here this end it is acetyl group, COH group. Again here it is hydrogen, and say suppose here again H is present. So these two amino acids, when they come together, from first amino acid, from the COH, the OH combines with the hydrogen from the NH2 groups and a water molecule is formed, and removal or dehydration of this water molecule takes place, forming a bond, and this bond which is formed between these two amino acid after removal of water molecule is called as a peptide bond. So two amino acids, they are interlinked by forming a peptide bond, and this peptide bond is formed because of dehydration, removal of water molecule. How is this water molecule formed? Acetyl group, it gives OH, and NH2 group gives the H. Now let us see further characters about proteins. They contain all naturally occurring 20 amino acids, or they may be made up of lesser amino acid. Recently, a 21st amino acid, selenocysteine, has been founded. This selenocysteine is found in only those organisms which are taking selenium diet. But basically, naturally, there are 20 amino acid. All may be present or few may be present in a protein. Proteins are extremely reactive and they're highly specific in behavior. They're amphoteric in nature. Amphoteric, they are acting as both acids as well as bases. They are strongly influenced by pH. Some proteins, they work very well at low pH, some works very well at high pH. So their behavior, what type of behavior the proteins will show, it is influenced by pH. Proteins are dipolar ions. Let us understand this concept. What is a dipolar ion and where it is acting as a dipolar? At isoelectric point. Now look at this first amino acid. You can see again carbon at one end, NH2, at other end acetyl group. Here there is amino group. Here there is some R group. This R group can be any R group, and here there is hydrogen. So generally this is an unionized form. But when it is ionized, you can see that NH3 gets one positive charge, so it is becoming a cation. COH, when it loses its electron, what is ion? Ion is formed by losing or gaining electron. So when COH loses electron, it forms a negatively charged, that is an ion. So but how are the positive and negative charges? Here there is only one positive charge, here there is one negative charge. So what is the net charge? One positive, one negative. They cancel each other, they are equal, and thus the net charge in ionic form is zero. Such type of ions, they are called as dipolar ions, where at isoelectric point the sum of the positive charges and the negative charges present are equal and the net charge is zero. Thus proteins are made up of dipolar ions having no charge, zero charge at isoelectric point. I hope you have understood this. Equal cation and anion are present, so net charge is zero. So, but if a protein, it has more basic amino acid, like lysine is there, arginine is there, lysine, arginine, these are basic amino acid, then that protein which has more basic amino acid, it behaves like a base, it behaves like a cation. Example, histone, positively charged. And therefore, histone, where is histone found? It is found in DNA, in the chromosomes. The chromosome basically it consists of uh, DNA which is coiled around histone protein. So I want to show you, suppose the structure of a chromatin network or chromosome. Chromatin network basically, this is histone protein, this is histone protein, this is histone protein which is an octamer, and it is a base, it is positively charged. Surrounding on this, there is a complete turn of one nucleic acid, then 3/4 turn, then there is a linker DNA, again one turn, again 3/4 turn, again linker DNA, and then alternate such type of histone proteins and DNA, it forms a chromatin network. So DNA is negatively charged, histone proteins are basic, they are positively charged. DNA is nucleic acid, acidic in nature. Histone is basic in nature. Histone is acting as a cation. And why it is able to act as a cation or positively charged? Because it has more basic amino acids like lysine, arginine. Do not forget this. Example, histone. But sometimes proteins are acidic in nature. They have more acidic amino acid. Then they are acidic proteins. For example, our blood, it contains proteins like albumin, globulin, fibrinogen, prothrombin. All these proteins are acidic proteins because they are present in our blood, and our blood is alkaline in nature. Our blood is basic in nature, and so the blood proteins are acidic in nature. Proteins are sometimes heteropolymers. Heteropolymers means what? They are not just chains of amino acid, always simple chains of amino acid. Sometimes along with chain of amino acid, they may have some metal ion, or they may have some other group attached. They may have one salt, or they may have some mineral present. So they are heteropolymers. Different types of constituents are present in a protein chain, not only amino acid. This separate thing other than the amino acid that is present in a protein, it is called as the prosthetic group. So in heteropolymers, they consist of these prosthetic groups. So students, I hope you have understood the characters of proteins. In next video, we will discuss about types of proteins. Thank you.